Hello, my name is Abhijit Ghosh. Of all the topics I studied this summer, the eukaryotic central dogma was the most intriguing. Therefore, it is the topic of this presentation. Please note that the prokaryotic central dogma is vastly different from the eukaryotic one. For example, a hairpin is not present in eukaryotic transcription termination, but it is in prokaryotic transcription termination. The central dogma is the process of creating a protein from DNA using mRNA as a middleman. It consists of two parts, transcription and translation. Transcription is the process of creating mRNA from a template DNA. mRNA acts as the middleman between DNA and protein. This middleman is necessary because there is a significantly higher chance of mutation of the DNA if it were translated directly. For example, if you needed to use one of Darwin's books, you wouldn't take home Darwin's original written copy. You would copy the text where the book is held and then take that copy with you to avoid damage to the original book. Transcription consists of four parts: initiation, elongation, termination, and post-transcriptional RNA processing. Transcription factors are the proteins that initiate transcription. TF stands for transcription factor. The 2 that follows it represents that the TFs help RNA polymerase II, the only polymerase used in eukaryotic transcription. Any letters or numbers following TF2 specify which TF that protein is. When the signal is sent that the transcription must occur, TF2D's Tata binding protein, or TBP, binds to the Tata box, a promoter about 30 nucleotides upstream or behind from the start site of the coding region that reads TATAWAAR. See the IUPAC notation for nucleotides. The TF2D bends the DNA at an obtuse angle. This signals TF2A and TF2B to bind immediately upstream and downstream to TF2D. TF2A stabilizes the binding of TBP to the Tata box, while TF2B will later help recruit RNA polymerase II. Then TF2F binds to RNA polymerase II and, using the help of TF2B and TBP, recruits RNA polymerase II onto the area downstream of TF2B. TF2F also prevents movement of RNA polymerase II unless the C terminal of RNA polymerase II is phosphatized, which means adding phosphate groups, which are 4 oxygen atoms surrounding a phosphate atom, to the C terminal. Currently, this is called the pre-initiation complex. Then TF2E binds to the RNA polymerase II. It recruits TF2H to the area downstream of the pre-initiation complex. TF2H is the largest transcription factor, with 9 subunits. Of that, 2 of them act as helicase and open the DNA, leading to the formation of open complex. Finally, the other 7 subunits act as a kinase and phosphatize the C terminal of RNA polymerase II. Then the process of elongation starts. Nucleotides attached with 3 phosphate groups are used in elongation. You may have heard of adenosine triphosphate. Adenosine is just an adenine with a ribose sugar attached. Another one of these is guanosine triphosphate. Another thing you need to know is that a hydroxyl group, which is 1 hydrogen and 1 oxygen atom, can do a nucleophilic attack to any triphosphate, resulting in only 1 phosphate group and the release of 2 phosphate groups. Finally, know that the 3' end of RNA, not DNA, only RNA, has an exposed hydroxyl group. So RNA polymerase grabs one of the triphosphates with the correct base, adenosine, guanosine, etc. It can detect the correct base because it has binding areas for all the bases, and the DNA base will bind to at least one of these binding areas. The RNA polymerase II can detect which one of its binding sites is bound to the nucleotide. Depending on which binding site it is, the RNA polymerase II knows the base of the DNA nucleotide. It places the nucleotide triphosphate on the first nucleotide. It then grabs the next nucleotide triphosphate and brings it close to the 3' hydroxyl group of the last RNA base while attaching it to the DNA, where the hydroxyl group does a nucleophilic attack to the triphosphate. When a hydroxyl group does a nucleophilic attack, it sticks to the 5' end of whatever it attacked. In this case, that binds the two nucleotides together with one phosphate group, which is the structure of RNA. This cycle is repeated for each nucleotide. The energy for all these reactions comes from the breaking down of the leftover two phosphates, which release a lot of energy when broken down. Certain transcription factors play a role in elongation of transcription. Transcription factor EB, or TFEB, acts as a kinase and phosphatizes any residues of the C-terminal that were not phosphatized by TF2H. This speeds up transcription as the GO signal for the RNA polymerase II gets amplified when more phosphate groups are added. TF2S speeds up RNA polymerase II when it becomes slow and does not allow for the pausing of transcription. During the elongation of transcription, cleavage and polyadenylation specificity factor, or CPSF, and cleavage stimulation factor, or CSTF, bind to the C-terminal tail of RNA polymerase II. In the end of the DNA, there is a polyadenylation identification sequence because it signals to start polyadenylation, which is adding the 3' poly A tail. When this sequence is transcribed and comes close to the C-terminal tail, then the CPSF and CSTF get attracted and bind to that sequence instead of the C-terminal tail of RNA polymerase II. Then a type of endonuclease, the actual enzyme name varies widely between species and even between cell types in one organism, and an endonuclease is an enzyme that cleaves or cuts RNA or DNA, and it's called the additional cleavage factor, binds where CPSF and CSTF are. It forces CSTF to unbind, and then it cleaves the mRNA right after the polyadenylation identification sequence. Finally, the additional cleavage factor disassociates with the mRNA. CPSF recruits poly A polymerase and then disassociates with the mRNA. RNA polymerase II disassociates, and the leftover mRNA downstream of the cleavage disintegrates. Post-transcriptional RNA processing primes the pre-mRNA to go out into the cytoplasm, where many enzymes could easily destroy the pre-mRNA. The post-transcriptional RNA processing consists of 5' capping, polyadenylation, which is adding of the poly A tail to the 3' end, and RNA splicing. 5' capping occurs in the beginning of elongation. During elongation, we discovered that the 3' hydroxyl group of nucleotide triphosphate molecules removes two 5' phosphate groups from each new nucleotide, leading to a nucleotide single phosphate. However, the first nucleotide had no previous nucleotide and therefore no previous hydroxyl group to remove the two phosphate groups. Therefore, there is always a triphosphate present at the end of 5' pre-mRNA. RNA triphosphatase cleaves the last phosphate group off of the 5' end of pre-mRNA. Then guanylyl transferase breaks a GTP into guanosine single phosphate and joins the 5' phosphate group from the mRNA and the 5' phosphate group of the GTP. Finally, methyl transferase adds a methyl group, which is carbon and 3 hydrogen, to the 7th nitrogen of the guanosine to prevent transcription of the guanosine. This completes cap 0. Methylation of the 6th nitrogen of the first base plus cap 0 is called cap 1, and methylation of the 6th nitrogen of the second base plus cap 1 is called cap 2. Cap 1 and cap 2 are rare, but cap 0 is common and almost universal. 3' polyadenylation is a continuation of the termination of transcription. First, Poly-A polymerase adds a strand of adenosines to the 3' end in the same way as elongation occurs. However, poly-A polymerase can do this without a template. After adding about 150 to 200 adenosine nucleotides, the poly-A polymerase dissociates with the mRNA. Finally, poly-A binding proteins get attracted to the open A nucleotides and bind to them. This is to prevent the nucleotides from forming hydrogen bonds with coding regions of the mRNA. RNA splicing is the removal of introns, or non-coding regions, from the mRNA. RNA splicing can occur in two ways: self-mediated splicing, no snRNPs, which I'll get to later, or spliceosome-mediated splicing, which requires snRNPs. The 5' end and 3' end of mRNA are always exons. Self-mediated splicing is splicing that does not require small nuclear ribonucleoproteins, which is snRNPs or SNRPs. SNRPs are complexes of proteins and small nuclear RNAs or snRNAs. Self-mediated splicing can occur in two ways: group 1 splicing and group 2 splicing. In group 1 splicing, the intron folds in such a way that a pocket is formed that can hold an external guanosine. The 3' hydroxyl group of the external guanosine is used to cleave the 5' end of the intron by doing a nucleophilic attack. The exposed 3' hydroxyl group of the first exon's 3' end is used to cleave the 3' end of the intron by doing a nucleophilic attack, resulting in the binding of the two exons into one strand. In group 2 splicing, there is a point called the branch point near the middle of the intron. Its sequence is just a single adenine. The 2' hydroxyl group located in both in DNA and RNA, and it is used because the 3' hydroxyl group has already been used in the nucleophilic attack during elongation of transcription of this nucleotide, is used to do a nucleophilic attack and cleave the 5' end of the intron, joining the 5' end of the intron to the middle and creating a lariat made of mRNA. Then the 3' exposed hydroxyl group on the 3' end of the first exon will do a nucleophilic attack and cleave the 3' end of the intron, joining the exons together. In spliceosome-mediated splicing, the U1 SNRP joins the 5' end of the intron. Then the U2 auxiliary factor, or U2AF, joins the 3' end of the intron. U2AF helps branch point binding protein, or BBP, to bind to the branch point. When this binds, the U2 SNRP displaces the BBP. This causes branch point to bulge out. Once this occurs, U2AF disassociates with the mRNA. U2 also recruits a complex of U6, U4, and U5 SNRPs. U6 displaces U1, and U4 disassociates with U6. U4 is only present to bind to U6 when not in use. Then U6 bends the mRNA into a U by binding to U2, making the branch point so close to the 5' end of the intron that the nucleophilic attack occurs. Once the exon is free, U5 will take the exon with it and bind to the exon 2, making the hydroxyl group of the exon 1 so close to the 3' end of the intron that the nucleophilic attack occurs that cleaves the 3' end of the intron. The mRNA is now ready to exit the nucleus and enter the cytoplasm to be translated. Translation is the creation of a protein using mRNA as a template. This is similar to using a copy of Darwin's book you took to do real crosses. Translation has 3 phases: initiation, elongation, and termination. EIF stands for Eukaryotic Translation Initiation Factor. Any letters or numbers after that signify which factor it is. In initiation of translation, Eukaryotic Translation Initiation Factor 1A, or EIF1A, binds to the A site on the small, lower subunit of a ribosome, called the 40S subunit, and blocks it. Then EIF1 binds to the E site and blocks it. EIF3 binds to the exit of the 40S subunit. EIF5 binds to the 40S subunit on top of EIF1. EIF5 stimulates EIF2 to loosely place a tRNA bound to a methionine amino acid into the P site. This is called the 43S pre-initiation complex. On the other hand, a complex is used to pull the mRNA is simultaneously being created. First, the EIF4F complex is recruited. This consists of EIF4E, which attaches to the 5' cap and pulls the mRNA to find the start codon, EIF4G, which attaches to the poly A tail, and EIF4A, which will hold EIF4B later. It joins to the mRNA, and EIF4B then EIF4B holds the mRNA coding sequence in a straight line during translation. The complex attaches to the 43S pre-initiation complex. The mRNA goes under EIF1A and EIF1, and the poly A tail loops under the 40S subunit. Then EIF4E pulls the mRNA until the start codon is aligned in the P site. Once this is found, the EIF2 will use energy from the GTP to bind the methionine tRNA to the start codon on the mRNA. EIF4G, EIF4A, EIF4E, EIF4B, EIF2, EIF5, EIF3, and EIF1 will dissociate from the complex. EIF5B will use energy from GTP to bring the large or top subunit called the 60S subunit and binds it to the 40S subunit. This binding will cause both EIF1A and EIF5B to dissociate from the complex. This is now called the ADS complex. Now it is time for elongation of translation. EEF stands for eukaryotic translation elongation factor. Any letters or numbers after that signify which factor it is. The first step in elongation is to read the codon of the A site of the ribosome. Then eukaryotic translation elongation factor 1A, EEF1A, uses energy from GDP to attach a tRNA with the correct codon to the mRNA. The EEF1A will dissociate from the ribosome, and a complex of EEF1B, EEF1G, and EEF1D will remove the spent GDP from EEF1A and will put in a new fresh GDP for more energy. The amino acid chain transfers from the P site tRNA to the A site tRNA, and then the mRNA shifts by one codon, dragging all the tRNA with it. If any tRNA are present in the E site during the shifting, then they will exit out of the ribosome and dissociate with the mRNA. The cycle repeats for each codon. ERF stands for eukaryotic transcription termination factor. Any letters or numbers after that signify which factor it is. When the stop codon is read, a tRNA is not recruited. Instead, eukaryotic translation termination factor 1, or ERF1, which is bound to ERF3, uses GTP to bind to the stop codon. Then ERF3 recruits RLI1 and a water molecule to the polypeptide chain. The water molecule and ERF1 help cleave the polypeptide chain. The exact way this occurs is still an area of active research. Then either ribosomal recycling factor or RLI1 uses ATP to disassemble the ADS complex into the 40S and 60S subunits. Finally, the protein has been created. That is a detailed overview of the central dogma. These are the resources I've used.